Hello students, good morning. Welcoming you back to my video where we'll be learning the next segment where we have left previously in our previous segment of the video where we have completed the lipids and we have started enzymes. And enzyme, what is enzyme and what are the properties of enzyme, we have learned that. Okay, so today we will see the factors affecting enzymes action. As we have learned previously that enzyme, it acts upon some substrate and then it converts that substrate into a product. Okay, so in detail we will learn in enzyme action how it converts the substrate into a product. Okay, so enzyme is just a mode which enhances the rate of reaction. Okay, it is a catalyst. Okay, so we will see this enzyme action which enhances the rate of reaction, how they are affected. Okay, in what way they are affected? Number one, it is temperature, which affects the enzyme action. Okay, temperature, with increase in temperature, the enzyme's activity also increases. Okay, with increase in temperature, the enzyme's activity also increases, and up to 40 degrees Celsius. Okay, up to 40 degrees Celsius, the rate of enzyme's activity is fast and it is increasing. Okay, because as the temperature increases, the enzyme's activity also increases. Okay, but as we rise the temperature up to 60 degree, and after that 60 degrees Celsius, if the temperature increases, like above 60 degree is 61, 62 or 70, in this temperature the enzyme's activity will decrease. Why so? Because due to high temperature the enzyme denatures. Denatures means it degrades. Okay, it degrades, it breaks down. The enzyme's nature, it does not work. Okay, in what it is supposed to do, it is supposed to work as a catalyst. That function it won't be able to do because it is degenerated. And why it is degenerated? Because of the high temperature. So up to this temperature the enzyme is working absolutely fine. It is working fast and it is enhancing the rate of reaction. But above that temperature, it decreases. The enzyme's activity decreases because the enzyme denatures. Denatures means it degrades, it breaks down. Okay, and at freezing point, enzyme becomes inactive. Okay, they don't work. Okay, so breaking down is different thing. Breaking down means where the enzyme denatures. Okay, and once the enzyme breaks down, it denatures, it will not function. So that we have seen when the temperature increases. But enzyme becomes inactive. It does not break down, but it will not function. Okay, it becomes inactive and the enzyme activity will stop. Okay, that is at the freezing point. So this was the effect of the temperature. Okay, how it affects the enzyme's activity. Number two is the pH. Okay, pH. Enzymes function best around neutral pH. At neutral pH. What is neutral pH? We know that pH value it increases from you have studied what pH means. pH means the concentration of the hydronium ion in the solution. Okay, or in a solution when the concentration of hydrogen ion. If the concentration of the hydrogen ion is it makes this concentration of hydrogen ion makes the solution acidic or basic. Okay, and also if it is neutral, then the solution becomes neutral. Okay, so comparison of this hydronium ion is done with the hydroxyl ion. Okay, hydroxyl ion. So hydroxyl ion and hydrogen ion. So if hydrogen ion is, the concentration is high, then the solution is known as acidic. If hydroxyl ion concentration is high, then the solution is known as basic. When the hydroxyl ion and hydrogen ion, their concentration is equal, it is known as neutral. In a solution, okay? In a solution, in a liquid solution. Now in a neutral, so on the basis of this concentration, we have a scale, pH scale, which ranges from 1 to 14. Okay? And in this 1 to 14 range, 0.7 refers to neutral, and below 7 it refers to acidic, and above 7 it is known as basic. You have studied this in chemistry, right? So at neutral, when a solution is neutral, the enzyme acts at its best. That is when the chemical reaction will be fast. If the enzyme is acting best, that means the chemical will react very fast. Okay? So in neutral solution, the enzyme acts best, but in highly acidic and basic solution, that is strong, okay, when the solution has a strong acid and basic solution, then enzymes again it denatures. Like how we have seen that in temperature, if the temperature is too high, then what happens? If it increases from 60 degrees Celsius, then the enzyme denatures. In the same manner, here also enzyme will denature if the solution is too acidic and too basic. Alright? So that is how pH acts. Number 3 is concentration of enzyme and substrate. Concentration of enzyme and substrate. If substrate concentration is increased, the rate of reaction also will increase. If substrate concentration increase, so rate of reaction also will increase. Okay? And the rate of enzyme action also increases up to certain limit. Up to certain limit. Alright, so there is a solution where you have added the substrate and where you have added the enzyme. Now the substrate, if you increase the amount of the substrate, the rate of reaction will also increase. The enzyme's activity will also increase. See, enzyme, it does activity on the basis of the substrate, presence of the substrate. If substrate is not there, enzyme also cannot do their function. Okay, so that we will see in case of enzyme action. So substrate, if we increase the amount of substrate, the enzyme's activity will also increase, but up to certain limit. You cannot keep on increasing, like in the same way, like how we have a temperature and pH. So everything works on a proper optimum value. If the temperature is optimum, enzyme acts at its best. If the pH is optimum, it will act its best. If the concentration of substrate is optimum, it will act best. So it should not cross the limit of the amount of substrate. Then the last factor is enzyme inhibitor. Now, inhibitors are those inhibits the enzyme's activity. Inhibits means to stop, to prohibit. Okay, prohibition. It inhibits the enzyme's action. How? As we have seen previously, enzyme has an active site here, okay, upon which the substrate is attached. This is substrate, this is enzyme. When these two gets attached, this site is known as active site, if you remember, right? This is active site. So now substrate will get attached here. So enzyme is very specific to the substrate. If the substrate is of this shape, it will not fit in here. Right? So enzyme does not act with this substrate, enzyme will act only with this substrate. So enzymes are very specific in nature, that we have learned. Now, what we are trying to say here is, there are other, like how substrate is a organic compound, like that there are other compounds which we call inhibitor. How inhibitors are? Inhibitors are somewhat similar to the substrate. So this also can fit in here. Okay, so if we add inhibitors here, we have added added inhibitor. So substrate now, a substrate which we had, it cannot fit in because already the inhibitor has fitted in this active site. Now this substrate cannot get converted into product. Okay, because enzyme is already been stopped. Enzyme is stopped by inhibitor because the active site has been occupied by inhibitor, whereas actually the active site should be occupied by substrate. But in this case, in case of inhibition, okay, enzyme inhibition. So inhibitor occupies the substrate space, a substrate space that we call active site. Okay, and how does they occupy that space? Because inhibitors are similar in shape with the substrate. Okay, so that's why they are known as competitive inhibitor. They compete with the substrate. Okay, they come first and they occupy this place, and that is how the substrate, they don't get this place, and then they cannot convert into product. Now why we do this process? To stop the enzyme reaction. Okay, so why this process happens in our body? To stop the reaction to happen. Okay, at times we don't want the we don't want the production of the product. We don't want to obtain the product. When our body don't need a product, that is when our body will release inhibitor, and that inhibitor will stop the enzyme action. Okay, so that is how these all factors, temperature, pH, and concentration of substrate and inhibitors, they affect the enzyme action. Okay. Now, to understand this better, we have a graph in your textbook from page number 156. You see, that graph explains us about the activation energy. So, progress of reaction, and this is potential energy. So, substrate, we know that substrate has a potential energy to do the enzyme action. Right? We know that substrate has a potential energy to do the reaction. Now, substrate will do the reaction in this manner. This is the potential energy graph line. Okay, and let me do the other line. Yes. Now see, what we are trying to understand from this graph. This peak represents the transition state. This peak, this peak and this peak represents the transition rate or transition state. Transition state. Okay? Transition of what? Substrate to convert into product. Substrate to convert into product. So you can see here, and this distance between the potential energy and the peak is the activation energy. So this line and this line, the distance between the potential energy and the peak is the activation energy. So the black line, black graph represents the transition state taking place without enzyme. So if enzyme is not there, okay, if enzyme is not there, so substrate will take lots of activation energy to transit means to convert into a product. It will take lots of activation energy and it will take lots of time. Okay, so the progress, you can see it takes lots of time to progress into a product. Now the red line, what does it show? Red line, the red graph shows that the transition of substrate to product in presence of enzyme. Okay, so red line shows in presence of enzyme. In presence of enzyme, substrate, it takes less activation energy to convert from substrate to product. Okay, it takes very less activation energy to convert from substrate to product. In very short duration of time, it will convert into a product. Whereas here in the black line you can see this is without enzyme. Sorry, this is not energy, enzyme, okay? Here also I am writing energy. It's enzyme, without enzyme. So without enzyme, substrate takes more activation energy to convert the product. So this is what this graph is trying to explain, that enzyme helps to convert substrate to product in a very short period of time with less activation energy, and without enzyme a substrate will convert into a product but with lots of activation energy and it will take lots of time. Okay, and from page number 157 you can see figure 9.7 where the graph is explaining this. It shows that the rate of reaction will increase up to optimum level. Then if the pH increases, if the pH keeps on increasing, then up to this optimum level, the rate of reaction, that means enzyme activity, okay? Enzyme activity, it will decrease. If the pH keeps on increasing after this optimum level, then the rate of action, the enzyme activity will decrease. The same way for temperature. Okay, up to optimum level, the temperature, enzyme activity. If the temperature keeps on increasing, the enzyme activity increases up to optimum level, optimum. After that, if temperature keeps on increasing, then the enzyme activity will decrease. Okay, and where the concentration of substrate, you can see the graph. If the concentration of substrate, okay, this is concentration of substrate and the enzyme activity or velocity of reaction. So if you increase the concentration of substance, the velocity of the reaction also will increase. It will keep on happening up to certain level, that is optimum limit. After that, no matter how much you are increasing the concentration of the substrate, the velocity remains the same. Okay, that is what the graph is showing. It will not change. If you are increasing the concentration here from 100 to 200 to 300, you are increasing. But what is happening to the velocity? It is remaining same. It is remaining same at this point. Suppose the velocity here is 10. So it is remaining 10 here, 10. Okay? No matter how much you are increasing. Okay? So that is graph figure 9.7. Graphical representation of the factors affecting enzyme activity. Okay? Now we will see the enzyme action. In the same page number 157, in page number 157 there are 4 steps given. Okay, 4 steps given of enzyme action, which explains the first step where enzyme combines with substrate. Okay, we know that if this is an enzyme, suppose I am drawing an enzyme here. So this is enzyme and substrate is of somewhat this shape. This is substrate. So this is the first step where this enzyme substrate will bind with the active site. Okay, here this is the active site. This is the active site. So substrate will bind here. Then what enzyme does? Enzyme changes the chemical nature of the substrate. Okay, so this is called ES complex. This is the second step, formation of ES complex, enzyme substrate complex. And when they are forming this ES complex, what enzyme do? It changes the nature of substrate. It changes the nature of substrate. And then third step, it will separate. Enzyme, when it separates now, with the substrate, this substrate has become already a product. Okay, when it detaches, let me do the structure here. When it separates, ENC separates, enzyme does not change. As we have studied from the property of enzyme, enzymes does not change. It remains the same like how it was before. But the substrate will change into a product. Now it is of different shape and size. Okay, because enzyme has acted upon this substrate and changed its nature. Okay, and that is how it releases the product. So stepwise this is mentioned in page number 157, so that you go through it. Okay, and then we will see the classification and nomenclature of enzyme. Types of enzyme we are seeing with different nature. Number 1, oxidoreductase is the name of an enzyme. Oxidoreductase, or it is also known as dehydrogenase. It means a compound, an oxidized compound, suppose S is oxidized, when it reacts with a reduced compound, okay, so it will lead to the formation of S get reduced, whereas S prime will get oxidized. So how this reaction can take place? By the help of enzyme oxidoreductase. So what does oxidoreductase enzyme do? It converts the oxidized compound into reduced compound and the reduced compound into oxidized compound. Okay, it does oxidation and reduction. Number 2, transferase. Transferase, it transfers the group. Suppose this is a compound attached with a group G. Okay, so S prime is another compound. So what does transferase enzyme do? It transfers this G group to the next compound. Here. So by the help of this reaction takes place. Hydrolase enzyme. So what hydrolase enzyme can do? Enzyme, it catalyzes hydrolysis. Okay, it hydrolyzes, it breaks down. Okay, it breaks down the bones. Okay, peptide bone, glycosidic bone, all these bones are broken down by this hydrolysis. Okay, catalyzing hydrolysis means breaking down this bone. Where did you see peptide bone? In protein, glycosidic in carbohydrate. So these bones are broken down. When we digest carbohydrate, so these all enzymes are used. When we digest protein, these all enzymes, to break down this peptide bone and glycosidic bone, these enzymes are used, hydrolysis. Then number 4 is lysis. Lysis is to transfer is catalyzed removal of group from the substrate. It removes the group. It does not transfer, it removes the group. For example, CNC has a group X and Y. So X and Y will be removed, okay, from CNC. Okay, so it will be removed from the compound. Suppose this is a compound and it has a group X and Y. So X and Y will be removed from CC, from this compound. So that is done by enzyme lysis. Next we have isomerase. What isomerase do? It helps in interconversion of the structure of the carbon compounds. Okay, optical geometric position. For example, if this is a compound, okay, now due to presence of isomerase, this compound will change the shape like this. Okay, this will change the shape like this. So to change the shape of the carbon chain, so you can see here the carbon chain is like this, here the carbon chain will be like this. That is the role of isomerase. The last one is ligase. Ligase is to join the, it links the two compounds or two elements together. Okay, two compounds together, two compounds. It links the two compounds. It joins the two compounds. Like lyase enzymes, they break the, they remove the group, and then hydrolysis, they break the compounds, whereas this ligase, they join the compounds. Okay, so and we are left with only one topic, that is metabolism with metabolic pathway. Okay, as we have studied earlier, metabolism is a biochemical reaction which takes place inside the cell. Okay, and it can be of two types, anabolic and catabolic. Okay, anabolic and catabolic. Anabolic is a constructive reaction which produces something like protein synthesis, and catabolic pathway is a destructive reaction which breaks down something such as glycolysis, where glucose breaks down. Okay, and this all leads to the production of inside the cell. So they produce some sort of substances such as metabolites, we call. So metabolites are the biological compounds which are found in plants or animals. So there are two types of metabolites, primary metabolites and secondary metabolites. Now primary metabolites are those biological compounds whose function are very clear, like amino acids, sugar, we know their function, they make protein, and protein make the structure of the body, and sugar provides you energy, it breaks down and provides you immediate energy for any kind of activity. So those are primary metabolites. Secondary metabolites are some of the products which are produced by animal or by plants, which their function we really don't know. For example, spices, why the plants they produce spices, and why they produce rubber, resin, drugs produced by plants. Their role in plants' body is not known. Okay, drugs which are produced by the plants, rubber which are produced by the plants, what role they play for the plant? Okay, they have important role in our life. We use the rubber, we use the drugs, and we use the resin in our life. We use the spices for our purpose. But what is their purpose for the plant? We don't know. So those kind of substances produced by plant body is known as secondary metabolite. Okay, so here we come to the conclusion, and the last part, which is known as cofactor. Cofactor is nothing but a cofactor are non-protein part of enzyme. Okay, we know that enzyme is protein, right? So along with enzyme there is a non-protein part which remains attached with the enzyme, so that is known as cofactor. Okay, so please go through it and read by yourself, and if you have any doubt in the cofactor, please let me know. Okay, thank you and have a good day ahead.